Hi, Dimitri. I'm fine.
Likewise.
It's great talking to you. How are you feeling?
I'm feeling fine, thank you.
So the genesis was really to do something about neglected tropical diseases in terms of making new medicines, and the impetus really came from conversation that Alan Fairlamb and I had back in 2002. So in some ways it's the 24th anniversary of the drug, or 21st anniversary of drug discovery unit. So back in the early noughties, I think it was fair to say that there was an extreme disinterest by the pharmaceutical industry in neglected tropical diseases. I'm not being down on them for this, it's just the way it was. There's no shareholder value in developing medicines for people who have no money to purchase them. Now it's a very different world where the pharmaceutical industry, or several of them, are playing incredibly important roles in this endeavour. But back then, really there was very little interest. And Alan and I were kind of wringing our hands about that, about how we could change that scenario. And we figured out that the only way to do that was really to in-source, bring in industrial pharma type activity into the university to do early stage drug discovery. So that was the impetus for setting up the drug discovery unit in the first place.
Well, initially it was really designed, or the intention was to try and tackle the unmet medical need for neglected tropical diseases, everything from African sleeping sickness and leishmaniasis through to malaria. That was the original vision, and that came out from a vision I put forward to the the Bill and Melinda Gates Foundation Great Neglected Diseases Grand Challenge. And so the concept had come from previous thinking of work I was doing with the WHO initially, where we'd identified key areas of neglect. Unfortunately, the Gates Foundation did not take this up as a grand challenge, and so then it came about that Mike and I decided to put this towards the Wellcome Trust as a proposal, and they were interested in it.
Well, there was absolutely no pathway to doing this whatsoever, so we had to sort of pave the pathway in front of us as we went. So yes, it could have failed at any and every juncture. So first of all, when we decided that we should do this, we had no laboratories in which to carry out drug discovery. We had no drug discovery equipment or facilities, and of course to get those two things you need to have funding. So our first roadblock, if you like, was, well, that's a great concept, but how are we going to achieve it? And almost always in life that boils down to where we're going to get the money from to do this. You can then fail in execution even if you get the money, but first of all you have to get the money. So at that point, Alan and I wrote to Sir Mark Walport, who at that time was the Director of the Wellcome Trust, and with a slightly cheeky letter that said, you know, dear Mark, Wellcome's been funding our fundamental discovery research for many, many years, for which we're very grateful, but we want to embark upon this new dimension of drug discovery, and we'd like to give Wellcome the first right of refusal in funding it. And I'm sure when Mark had finished laughing at that, he was gracious enough to invite Alan and I down to Wellcome to discuss it. And so we went down, and after some discussions were invited to put together a large sort of big program grant to get the whole thing started. And then the ball was in our court to write that, and then Wellcome assembled a very specialist review panel with people from the pharmaceutical industry to kind of kick its tires and see whether it was worth funding. And we were very fortunate that Wellcome funded in 2005, actually the grant came through, 8.6 million, which back then was a lot of money, to give it a go basically. and we established the DDU in early 2006, January 2006. So that's the history of it, really, in terms of the initiation of the drug discovery unit. Yeah.
Well, there were certainly a lot of concerns as to why suddenly we wanted to go it alone. And so I did send a lot of correspondence to the Wellcome Trust, showing that I'd been trying to work with a major pharmaceutical company for many, many years, and illustrating how I was always at the tail end of any decisions. So when mergers and acquisitions took place in pharmaceutical companies, projects would be cancelled at the last minute. When there was political arguments about costing of HIV/AIDS drugs, and pharmaceutical companies got upset about pressures that they were being put on. Again, projects on neglected tropical diseases, of course, were cut. So it was very frustrating, and I had a huge batch of email correspondence which I sent to the Trust saying, this is why we want to try and tackle this problem ourselves. So that's how it came about, basically.
Well, I think there was a deep understanding by people like Mark Walport and other people at the Wellcome Trust that indeed there was a hole in the market, that there was a broken market, in that people in countries with neglected tropical diseases or endemic, where they're endemic, were never going to get any new medicines unless there was a different paradigm to try to get to them. And it wasn't going to happen through the activities of the pharmaceutical industry itself, because there wasn't the incentive to do so. And so that was recognised by the funding agency, in this case Wellcome. It was also recognised by Gates around about the same time in other areas as well. And so the charitable sector, the third sector, has played an incredibly important role in catalysing the creation of new medicines, which are then partnered with the pharmaceutical industry and taken to the clinic. So, but there was just this big gap, this void of nothing, no pipeline, basically.
Well, the original vision was target-based approach, in the sense that about a decade previously, a colleague of mine, C.C. Wang, had published an influential paper saying that really there were certain criteria that should be met for drug targets. And one of those was genetic validation. So myself and other colleagues had gone through a lot of effort to validate and show that targets were essential. So we had a lot of targets. We had three-dimensional structures for some of them. We knew their role in the biological processes of the parasites, and so we were well grounded in a target-based approach with what we thought were validated targets at the time.
Key milestones, well, first of all, we had to build some labs to put drug discovery in, and so at that time, together with Sir Philip Cohen, I was fundraising for the Sir James Black building, and the ground floor of that was dedicated for setting up the initial drug discovery unit, so we had to raise that money. That was key. So you have to have somewhere to put activity. Second one was the raising of the first 8.6 million to get an inoculum drug discovery unit team in place. That was really the second major milestone. And the third one really was engaging with the wider scientific community to make sure that we were taking in the best possible drug targets and drug screening methodologies to get it going. So this was never an extension of my lab and Alan's lab or anything like that. We realized we were creating something that had its own distinct identity, should have its own leadership and its own criteria?
Well, the first milestone was actually 1 month in, where we had to have an appointment of a director of the drug discovery unit, because although I'd done decades worth of service to WHO drug discovery committees and so on, Mike and I had no full experience in drug discovery, so we recognised that from the outset, and therefore had to find a suitable director of drug discovery who could manage the chemistry, in particular aspects of drug discovery, and that led to the appointment of Paul Wyatt early on. So that was the first milestone. He then asked us about the different targets we had and the biological models we had, coming from a cancer background, had no experience on neglected tropical diseases. And he listened to what we had to say, and he said, well, actually I think you should focus on African sleeping sickness. You've got the best validated targets there. You've got a robust in vitro and in vivo models. Let's focus on that. And we worked through a number of targets in the first 5-year program, but one particularly led to a partial success, and that was an inhibitor against N-myristoyl transferase in the parasites. Unfortunately, although we'd got this to the point where it could be a preclinical candidate, we were informed by changes in policy at WHO that they were no longer seeking a drug that would merely cure the first stage of sleeping sickness. It had to be able to cure both stages, and we had focused on a different product profile, and then suddenly they said, no, we don't want that product profile. So that was a little discouraging, and indeed we did continue to try and meet the requirements for a single drug to cure both stages of the disease, but the challenge was getting the drug into the brain, crossing the blood-brain barrier. But we did have proof of concept, and the interesting element about that work was that it was published in a high-impact journal under a very strict reviewing process. And that was a key event in the sense that pharmaceutical companies now knew that this was a professional organisation that was able to carry out the drug discovery process. And I think that was one of the key events that came about in the first 5-year programme, such that in the second 5-year programme, we were encouraged to bring on board a pharmaceutical company as a partner, and that was definitely a game changer at that point. So those were definitely key events. And another one I can think of had to do with the importance of pharmacology in the drug discovery process. In the initial plan, we proposed to outsource this to different CROs to carry out the pharmacology. But we realised this was impractical and very expensive, and so we recruited Kevin Reid, now Professor Kevin Reid, to head up the drug metabolism and pharmacokinetic aspects of the process. And I think that was another key event that really helped us formulate the drug discovery unit as it stands today.
I think what made the biggest difference was one of the first people that we appointed was Julie Frearson, and she came from Biofocus, and she was an expert in high throughput screening of compounds. So obviously if you're going to do drug discovery, whether you're going from phenotypic screens of finding compounds that kill the bugs themselves, or target-based screens where you have a recombinant enzyme which you think will be a good drug target, and you screen against that. Regardless of which way you go, you need to have competency in that screening biology. And so bringing Julie Frearson in at a very early stage from industry, from Biofocus, was a very, very key moment. At the same time, Ian Gilbert joined us to be head of medicinal chemistry, and then to come in as basically the head of the drug discovery unit. Shortly thereafter was Paul Wyatt, who came from via GSK and Aztecs Therapeutics, came to be overall sort of director, if you like, of the drug discovery unit, and he brought that sort of industrial rigor and viewpoint across the whole thing, together with Julie. So those were really incredibly important appointments, and if we hadn't had those three, and Ian, Paul and Julie initiating the drug discovery unit, I don't think it would have grown in the way that it has. So the credit really belongs to them. Julie subsequently left, got a very high profile job in the States. She's now Senior Vice President for Charles River Laboratories. And David Gray came in to take her role of Head of Biology. And along the way, Paul made a really, really key decision, which was to not outsource our pharmacokinetics analysis to companies, but to bring it in-house. And he appointed Kevin Reed from GSK to lead up our so-called DMPK, Distribution, Metabolism, Pharmacokinetics team. And that was an absolutely key decision point, actually. And I hadn't appreciated before how much better and faster and more effective a drug discovery campaign can be by having your DMPK in-house rather than out-of-house.
Yes. Clearly, over 5 years, we went through a large number of molecular targets with varying degrees of success, but ultimately, many of these targets just fell by the wayside, N-myristoyl-transferase being the one that was successful. And Paul Wyatt persuaded us that the next step was to move to what's called phenotypic screening, where you take an agnostic approach to drug discovery, where you take drug-like chemical libraries and you test the individual compounds against the whole parasite. And your phenotype you're looking for is a dead parasite, okay? And that led to whole new areas of chemical matter which had not been explored before, but ran into the second problem of how do they work? And so the second part of that success was the establishment of a mode of action team, initially in my group, but subsequently expanded and extremely successful under the leadership of Susan Wyllie. And I think that was another transformational step, such that we could go from compound to target to structure-guided drug discovery. And I think that was another important event, which all of these features actually together make the drug discovery unit quite unique in an academic setting. There are very few equivalents, certainly in the UK, that could match the capabilities that we have here.
I'll do two very quickly.
Sure.
So I think, so the first is the first one really, which was in the early years of the DDU, we took in a molecular drug target for African sleeping sickness from the University of York, from our friend Debbie Smith. It was NMT, N-myristoyl transferase, and that was a textbook piece of science. It was lovely, and it was all published as a beautiful big article in Nature in the end, of taking a recombinant enzyme which is of known essentiality in the parasite, doing a high throughput chemical screen, finding start points, doing elegant medicinal chemistry, and coming up with a very, very high quality molecule with excellent pharmacokinetics, excellent solubility, excellent potency, etc., etc. And indeed it was a great molecule to cure stage 1 African sleeping sickness. That's before it gets into the brain. However, it failed because it wasn't, it wouldn't get into the brain and cure the stage 2 African sleeping sickness. So whilst it was a beautiful piece of translational science, ultimately it failed for neglected tropical diseases. But because David Gray and Paul were convinced they had an asset of great value, they ended up partnering it with Luc Bertinome in Canada, and it's now, it was transferred, the IP was transferred to a spin-out, well, a startup company in Canada, and it's now in phase 2 clinical trials for acute myeloid leukemia. So that's a great example of creating a really high value asset, fails for its primary objective, but it's re-tasked for a second one. And that's very exciting and looks very promising. Keep our fingers crossed for that. The second one, I'll be even briefer, was of course the Cabamiquine story in malaria, where actually it was Julie Frearson who was keen to initiate something in malaria, and they ran a very small screen, only about 5,000 or 6,000 molecules, kinase inhibitor panel, against the parasite itself, so phenotypic. And a very talented, but quite small team optimized those, one of those hit series to what is now called Cabamiquine. And turns out it doesn't hit any kinases whatsoever. Right, so it morphed from being starting from a kinase inhibitor set to being a protein synthesis inhibitor. But you know, that's just completed phase 2 clinical trials very successfully in Africa, and that's the one we've got all of our fingers crossed for there. And Beatriz Baragana did a fantastic job on the medicinal chemistry, and of course she now directs the malaria portfolio within the DDU really excellently. So those are two stories both of which I'm, you know, very proud of, to see what the DDU, not me, but what the DDU have done with the facilities and assets that they have.
One in particular would be the anti-malarial program, which started off with a very small focused library of protein kinase inhibitors, some of which were found to have activity against the malaria parasite, and I think they grouped into about 9 different subcategories. And as we went through them, several of them fell by the wayside because there seemed to be no way forward in terms of selectivity. And now we were engaged not only with a pharmaceutical company for this kind of research, but also with a public-private partnership called Medicines for Malaria venture. And they were acting as advisors on the different chemical matter we were working on. And so we ended up with one series that looked very promising, that did not overlap with other projects that this virtual pharma MMV was operating with other institutions. And the next key element of that was discovering that these protein kinase inhibitors were not acting through inhibition of protein kinases, but actually affecting protein synthesis. And the compound then, the compound series came to an optimal condition within about 18 months, very rapid indeed. and was then taken on by a major pharmaceutical company and is currently in phase 3 clinical trials. So that is a real success story against a major neglected tropical disease affecting the poorest of the poor globally. So I think we're very honoured with the success that has come from that particular project.
I think it's very open to collaboration, and indeed all of the programs, whether it's malaria or whether it's kinetoplastic diseases, which Manu de Rycker runs that portfolio incredibly well. That's all done with GSK, in collaboration with GSK, has been for 15 years, for example. Produced two candidate drugs which are just completing phase 1 clinical trials. Fantastic piece of work. The original sleeping sickness program was done in collaboration with York and the Structural Genomics Consortium in Toronto, for example. So at every stage and every program there are multiple collaborators, both national and international collaborators, and I think that's the point. The DDU has a philosophy and a culture of getting the job done and not worrying about where the kudos goes so much, right?
Yes, well, I mean, the method that's used is to form a team structure where you have a diverse range of expertises all working together on particular projects. I'm not sure that that is necessarily unique in the pharmaceutical industry, but it certainly has stood us in a good stead within the DDU. So having a blend of disciplines, people like myself who can act as an interpreter sometimes when biological questions come up or complex enzymological questions come up. Those are my areas of expertise. Pulling all these things together and working as a team is very important. And the other thing I think about teamwork is you don't have room for too many big egos. You all have to learn to work together and defer to the experts in their area, and you have to learn to let go. You have to let go of your favourite target when the chemistry's telling you it's not going anywhere, or the biology's telling you it's not going anywhere. So you have to be brutally honest with yourself and accept that your favourite target is not going to make it. And indeed, that's a lesson well learned by Mike and myself in terms of drug discovery. Yeah. Well, so, when Alan and I set it up, of course, what we had in mind, because we're both biochemists, is target-based drug discovery. In other words, people like Alan, people like myself, people like Debbie Smith in York, etc., could come up with a single enzyme of such essentiality to the parasite that we would drive the program around finding inhibitors of that specific enzyme. And then it'll be a beautiful story. We found, we learnt this about the parasites, and therefore we selected this magic target, and we found, made drugs against it, and lo and behold, we've cured the disease. And we didn't do that in the end. And so we pivoted to what's called phenotypic screening, which is, well, let's just find molecules that kill the parasite and then optimize those, and then figure out what they're hitting afterwards. So, and of course, that was something that Alan had been involved in for many, many years, in drug mode of action. And of course, his colleague Susan Wyllie now drives the drug mode of action team, which is one of the best in the world, in my opinion, at what it does. And so that's been the pivot from picking a winner to letting the biology select the winner for you. That's the pivot that's really made the difference. Yes, well, I've touched on some of those. I would say that the switch to phenotypic screening required the mode of action team being created, and under Susan Wyllie's guidance, and the fact that she's been able to deconvolute, I think, over 60 drug targets now, not just in-house but externally, which is very important for making sure that we have the right kind of portfolio globally for drug discovery projects. Then I've mentioned before bringing in-house pharmacology, because ultimately you can make the most fantastically potent molecule that acts in the test tube but is immediately chewed up in the body or is not absorbed in the body. So I think the presence of Kevin Reed's team has been transformative. And then more latterly from Kevin's work has been the development of a humanised mouse model for many of these diseases that we work on. And I think the importance there is that compounds which are rapidly destroyed by mouse metabolism would normally be discarded because you have no way forward in development. But with the humanised mouse model, you can then find that the predictive value of those this animal model is extremely helpful in deciding what would be most suitable for human patients.
Well, it should be known for actually having made some new medicines for tropical infectious diseases, maybe a new anti-malarial, maybe a new anti-leishmania drug, maybe a new tuberculosis drug. The work that Lorna Cleghorn's been doing is fantastic in that area. So there's early stage drug discovery. Our job is to get to a preclinical candidate and pass it on to the downstream product development partners. But then there's lots of places where it can fall over in phase 1 trials. Maybe it's not safe enough. In phase 2, maybe it's not efficacious enough. In phase 3, maybe it causes idiosyncratic toxicity you hadn't spotted before. All sorts of reasons. And then cost of goods, maybe it proves to be too expensive for that market, etc. So lots and lots of places it can fail. But I think what I would really be proud of in the next 5 years or so is if any one, or hopefully more than one, of our prototypic medicines actually gets registered and starts being distributed to patients. Because otherwise, what's the point of drug discovery?
Well, I would hope that it's known for several successes or contributing to several new treatments for neglected tropical diseases. It should hopefully also be known for showing that focusing on initially on parasitic diseases, you can actually come up with treatments for cancer cells. I didn't mention, but the N-myristoyl transferase compound series turned out to have value against a rare form of blood leukemia, which has now got accelerated approval through the FDA process. And that came from working on a parasite target. And the interesting point about that was we never got selectivity between the parasite human NMT and the parasite and the human NMT. Very little selectivity there. The selectivity is in the downstream biological process that the NMT was involved in. And remarkably, that's how you can sometimes get selectivity. And so it was an interesting lesson. So, you know, hopefully we will be remembered looking back that, yes, we tackled a number of unmet medical need for diseases of poverty, and we're also able to contribute towards the development of new therapeutics in other areas, what one might call orphan diseases, where again pharmaceutical companies are reluctant to invest early on, that we can bring chemical matter and pharmacological validation of a particular process, biological process, that could lead to modification of a rare disease as well. So I think those are the two elements I would like to see the Drug Discovery Unit recognised for.
It's always funding, right? Funding agencies change their priorities, and it's absolutely their prerogative to do so, so I'm not complaining here at all. But, you know, a good example would be, at the moment, the Chagas disease program that Manu de Rycker is leading. I mean, he's done a fantastic job. He and the whole team and the GSK co-team have done a fantastic job and have just gotten to a position where, you know, they're just about to get a preclinical candidate over the line, but the funding, world funding environment has changed, and now there isn't funding to do that final little bit, if you see what I mean. Well, obviously we're looking for it at the moment. So things can go off the rails for what are ultimately spurious reasons. Some other third party or second party funder, in this case, we call them the second party because rather than a third party, simply changes their policy or priority, as I say, which is totally their prerogative to do so. But then you're left high and dry with an asset which is almost there, and it's like, well, anybody want to help us get this over the line? So that's one risk which is always there.
Well, the biggest risk always is loss of reputation of the DDU, loss of funding for the DDU, and loss of vision and leadership for the DDU. So I think there are three major risks that spring to mind. In terms of future success, I suspect that the increasing use of artificial intelligence or machine learning should have a transformative effect on the way we operate within the Drug Discovery Unit.
Yeah, I think, you know, when the DDU was set up, and we were sort of designing its overall shape, I always saw it as what I could refer to as a translational engine. You can put in top quality biology at this end and produce top quality preclinical candidate molecules at the other end, and everything in between. And that therefore it wasn't just built solely for neglected infectious diseases, although that was totally the imperative for setting it up in the first place. And I always thought, well, it should also be looking at other areas of unmet medical need, oncology, plenty of cancers which are not treatable at the moment, neurodegenerative diseases, for example, but not copying what's going on in pharma, but actually looking for innovative targets in those disease areas which pharma are not looking at. Things which pharma often describe as too early, too risky, which is quite a lot of things. And moving them forward and de-risking them and partnering them with industry further downstream, because if you've de-risked something and you take the data package to pharma, then they may take it on at that point. Or you might create an asset which can then be spun out into a spin-out or startup company to take it towards the market. And it's been slower than I anticipated to build up that activity, and we have, of course, the innovative targets portfolio which does exactly that. And I was quite surprised. I thought we'd move quite quickly over the years to go to about 50-50 in both areas, but we're still over 60% in infectious diseases and less than 40%, or they're about 40% in innovative targets. And I think over the next few years, I would like to still see that balance shift a little bit towards the innovative targets portfolio, because for one thing, it could create an income stream to fuel the virtuous cycle, including benefiting neglected infectious diseases. So I'd like to see that happen.
The next chapter for drug discovery. Well, I think it depends on identification of new areas of unmet need, where we feel that the drug discovery unit would be capable of making a realistic contribution. And in many ways, that actually depends on the structure of life sciences itself in Dundee, because I'm a great believer that we have far more success when we have in-house scientific expertise and knowledge of a particular disease process that one wants to work on. So without being prescriptive, I would say, generally speaking, if we have that combination of fundamental scientific knowledge and extreme drug discovery expertise, and bringing those two together, that's really the way I would like to see the drug discovery unit progressing in the future.
Well, I don't know whether you can see that far in the future, Dimitri. I think it's really important. So you have to have a balance between action and directed activity. In other words, if you're running the malaria portfolio, you have to have your focus on making anti-malarials, absolutely no question. But you also mustn't lose sight of scientific innovations which are happening all the time from all different areas of bioscience, to make sure that you're always using the sharpest tools to get to the best possible drug. The patients deserve the best technology directed onto their problem to get the best drugs. So that's really important. But there's a trade-off there between focus and innovation, access and adoption of innovation, because both take energy and time and money. So getting that balance right is quite difficult. And like everything, there's a hysteresis, an oscillation in a way. Sometimes, you know, if I look back historically, there might have been moments where perhaps we were putting even slightly too much into the innovation area, and other times when we've been putting too much in the vertical portfolio. Let's get to a medicine portfolio, a medicines product, and getting that balance right between the cross-cutting technology and the vertical get a job done thing is difficult. I mean, I have a saying which kind of argues against it, but the other balance is not to overdo the innovation thing. I always say never let enabling technology slow you down. Sometimes you don't want to be in front of the wave, you want to be just on the crest of the wave and letting other people, there's a big world out there, develop technology. But once a technology is mature and really has shown its usability, then you must really adopt it and apply it. Plate-based chemistry would be a good example of something that's been completely adopted here now, but there was it took a while to get it adopted.
20 years from now, you are looking very far into the future. Well, advice I would give them. I think that 20 years from now, we may indeed have huge changes in the way we carry out drug discovery. I can only sort of take a parallel look at what has occurred over the 20 years plus of drug discovery activity that I've been involved in. And you know, throughout my career, drug discovery research used to be that you had an animal model and chemists made sufficient compound to test on an animal model. That was the way it was done in the 1960s and 70s. Then along came genomics and the developments in cell culture that allowed us to miniaturize these screens. Then along came combinatorial chemistry. Suddenly that was the new vogue that you would just throw millions of compounds at a problem and that would sort things out. And then we discovered that wasn't quite the case, and then it was pharmacology came in. So all of these elements have been developed. So to look into the future is, I think, is asking a lot. It may be that in 20 years' time, perhaps we have a complete in silico model of how a cell works and how an organ, a tissue, an organ, and a whole human being operates. We could have a model of that nature, and perhaps more and more artificial intelligence is used to carry out the early phases of drug discovery before you even make a molecule. Like that's maybe 50 years in the future, but if you want to think big, yeah, that kind of in silico challenge would be absolutely incredible if one could come up with suitable models of that type. And indeed, who knows? You know, it may be that in the future, you want a pharmaceutical. You may go up to a machine and punch a few buttons, and the chemistry's carried out in front of you, and you're provided with the tablet there and then. Who knows? You know, those are science fiction. Now you're taking me into my hero, biochemist Asimov, thinking about, you know, way in the future what might occur. Yes, you need to know your limitations. You need to respect the opinion of others. You need to learn how to work together as a team. And you also have to have a huge amount of determination. Throughout my career, I've never lost sight of what I wanted to do, and that was to discover new treatments for tropical diseases, or at least to contribute to them early on in my career. And it was not about chasing high-impact publications. That's so easy to do, but it might not lead to a new treatment for a tropical disease. So keep your eye very focused on what you want to do. If you have that laser-like vision and determination, I think that is one of the key things you need to be successful in drug discovery.
Thank you so much, Mike. It's always great talking to you.
Okay, thanks, Dimitri.
That one got you?
Yes, yes,
Good,
yes.
excellent.